Hello, my name is Zachary Fralish.
I'm Ashley Norberg.
And I'm Nikita Pearson. And today we're presenting a research paper called GAD45A Gene Silencing by RNAi Promotes Cell Proliferation and Inhibits Apoptosis and Senescence in Skin Squamous Cell Carcinoma through the p53 signaling pathway. Here is a redacted abstract. I'll give you a couple of seconds so that you can review it quickly, but this is also available in our handout. Okay, so the model organism used in this research were called nude mice, or also called athymic mice, because they are hairless mice and they don't have a thymus. Without a thymus, these mice are immunocompromised and unable to produce T cells. The hairlessness of these mice allows for the consistent absorption of UV radiation, or UVR, which is necessary for this research to take place. And the weakened immune system is taken advantage of because it allows researchers to transplant skin or tumor grafts onto or into the mice with little to no rejection from their immune systems. However, this requires completely sterile environments because they are easily infected by pathogens that can be fatal in nude mice, even if they are not fatal, or even if they are commensal in normal mice or humans. In the case of this study, the researchers injected genetically modified cell cultures under the skin of the mice. There were 6 groups of mice in total. The first group were 8 mice that received no treatment and served as the control group throughout the entire experiment. And then the other 40 mice were continuously irradiated by solar lamps that emitted UVR for 20 weeks in 12-hour light-dark cycles. After the 20 weeks, the 40 were split into 5 more groups, each injected with skin squamous cell carcinoma cell lines that were either not transfected, which they denoted as blank, transfected with an empty vector, which served as their negative control, which is abbreviated as NC, or transfected with siRNA GAD45A, which they abbreviated to siGAD45A, Nutlin-3, which is an activator of the p53 pathway, or siGAD45A plus Nutlin-3.
So as Nikita was saying, the protein that this study focused on is the GAD45A, which stands for Growth, Arrest, and DNA Damage Inducible 45 Alpha. This is an RNA binding protein, and it is usually rapidly induced by the stress from cell apoptosis and the growth arrest through both the p53 dependent and independent pathways. GAD45 protects the hematopoietic cells against the UV-induced apoptosis. This study focuses on the p53 pathway, which responds to help regulate genes that induce apoptosis, the cell cycle arrest, DNA repair, and senescence, which is a growth inhibiting state of the cell cycle. If you think back to the cell cycle, there are checkpoints that the cell goes through before it can proceed to the next step in the cycle. Failure of a cell to control its growth, especially containing damaged DNA, can lead to camerous tumor growths. So, some of the mice were transfected with siGAD45A to test the cell proliferation, apoptosis, and senescence through the p53 signaling pathway following the silencing of the GAD45A. This was done using RNAi, our technique of the week. RNA interference, RNAi, is the process by which the translation of a protein is prevented by selective degradation of its encoded mRNA. RNA can be used in a few different ways to manipulate the expression of a gene in an organism. By manipulating genes, we're able to test the roles of the genes and how their manipulation can affect the organism. This study used small interfering RNA, which is created by utilizing a double-stranded or hairpin structure of the RNA to form a RISC complex. This can bind and signal the degradation of the mRNA and lead to the silencing of a target gene.
So ultimately, the investigation we are presenting seeks to figure out how GAD45A is involved within skin squamous cell carcinoma. The first step in this is through the silencing of GAD45A through RNAi in the nude mouse model of SSC as depicted in the top left corner. Then, researchers analyzed resultant tumor progression, cellular properties, and protein expression through physical measurements of induced tumors in the mice, and then performed a variety of assays including MTT, ELISA, and immunohistochemistry to determine cellular properties.
The first graph that we would like to talk about are two bar graphs, which show the skin tumor size and the weight of each group. The groups with the transfected silence gene had the largest size of tumor growth in both size and weight, as you can see here and here. You can see the blank and negative controls and the SI-GAD45A plus the Nutlin3 here showed the same size and weight of tumor growth, while the Nutlin3 alone showed the smallest tumor growth, while the Nutlin3 showed also the smallest weight growth. They classified the blank, negative control, Nutlin3, and the silence GAD45A plus Nutlin3 combo to be mild degree tumor growth, while the SI-GAD45A was severe degree growth. This led them to believe that the SI-GAD45 induced tumor growth, while the Nutlin3 alone inhibited tumor growth.
In order to analyze the viability of cells through skin squamous cell carcinoma after silencing of GAD45A by RNAi, MTT assays were conducted. To do this, tumor cells were extracted from diseased nude mice and seated in 96 well plates, and assays were run in 8-fold at various time points. Following the addition of the MTT solution and fluorescence detection at 490 nanometers, it was seen that transfection with SI-GAD45A led to increased rates of cellular proliferation at all time periods analyzed. Additionally, it was seen that Nutlin3 reduced cellular viability at all time points analyzed. Nutlin small molecules occupy the p53 binding pocket of MDM2 and effectively disrupt the p53-MDM2 interactions that lead to the activation of the p53 pathway in the p53 wild-type cells. Inhibiting this interaction between MDM2 and p53 stabilizes p53 and is thought to selectively induce senescence within the cancer cells. There was no significant difference in cell viability between the control group and the group with both SI-GAD45A and Nutlin3. This suggests that the effects of SI-GAD45A counteract the effects of Nutlin3. So ultimately, it was seen that cells that were transfected with SI-GAD45A exhibited promoted viability, underlying the importance of the presence of GAD45A to limit cancerous growth.
So the last two figures that we want to show you depict concentrations of the 4 inflammatory factors and vascular endothelial growth factor, which they abbreviated to VEGF. And this is a growth factor that is commonly upregulated in tumor cells. And all of these were targeted in this study and analyzed using an ELISA. In both figures, we see that when compared to the normal group, all inflammatory factors and VEGF were present in statistically significantly increased levels, which is denoted by the pound sign or the hashtag mark. We also see that when compared to the blank group, all inflammatory factors and VEGF were statistically significantly increased in only the SI-GAD45A group of mice, but all inflammatory factors were significantly reduced.
Ultimately, this paper suggests that GAD45A has potential as a viable therapeutic tool in the treatment of skin squamous cell carcinoma through showing increased spread and livelihood of these cells after the use of RNAi to silence the GAD45A gene within these cells. This claim is supported through a measured increase in the weight, size, and number of tumors within the SI-GAD45A group. Additionally, the presence of Nutlin-3 counteracted the effects of GAD45A silencing, which underlines the importance of the p53 signaling pathway and the possible therapeutic effects of GAD45A. Altogether, the results revealed that GAD45A silencing increases tumor cell proliferation and reduces apoptosis and senescence through the p53 signaling pathway in the skin squamous cell carcinoma model. Within the drug development process depicted on the bottom of this slide, this paper is in the realm of the early stages, but offers promising results. Further investigations are necessary to more fully understand the mechanisms through which GAD45A reduces tumor cell proliferation. Additionally, tests with increased presence of GAD45A should be performed to see if this helps reduce tumor proliferation.
And these are our references.